don't know.
Yeah, you probably just have a lot of space.
Huh? Yeah, it doesn't. No. It works. Usually it works. Sometimes it doesn't. Okay, we are back from spring break. I hope you guys all had fun. I was rear-ended on Monday, totaled my car, but I ended up walking away from a mangled wreck. Unfortunately, I was very sore for a day or two, so I kind of about half of my spring break was spent lying, being pitiful. But I ended up having a good one. I did a boatload of mountain biking, so I can't complain. So we're over the halfway mark. If you guys look at your syllabi, that's a good thing, right? Let's see, what do we have coming up? We've got your second lecture exam on Tuesday, and a significant amount of material in that exam will actually come from today's lecture. So this is definitely a very important lecture. And after this lecture, I'll try to wrap this up a little early if I can, and then I can do kind of a little review, give you guys an idea of what to expect. And then we have got, let's see here, the withdrawal date for W is coming up next week as well, I believe. So that's something to keep in mind if you've been having significant issues on the last few exams. Since, let's see, we've had two exams up to this point, right? We've had a lecture exam, we've had a lab exam. So at this point, you should have a reasonable idea of kind of how you're doing. And of course, all of your midterm grades got posted. I did do a complete review of the lab exam. Hopefully, I don't know if you guys noticed that on Canvas or not, but I have 2 videos up there for you guys. One video for all the models, the disarticulated and articulated bones, and then a second video with histology slides. Actually, I just took pictures of the exact slides that you guys had, so please review those if you want, particularly if you struggled on the lab exam. There was a fair amount of struggling on the histology, which was interesting because I actually chose slides that were a bit less ambiguous. But on the other hand, people seem to do really good on the bones, which sometimes people really struggle when it comes to bones. And you guys, for the most part, did pretty good on bones, but struggle a little bit on the histology slides. So please use those resources, and I'll keep them up the rest of the semester. So that's not a problem. I'll try to do something similar for your second lab exam, which is coming up not for a while, it'll be in April, so you don't have to worry about that too much. Okay, so let's go ahead and jump right in. What I want to do is I want to start off with a quick review, since I'm sure you guys have all been studying. Great. You're up on your reading, and you have a very robust memory of all the concepts we've covered up to this point. Am I correct in saying that? No, I'm not. No, I'm not. I'm completely incorrect in saying that. Okay, I know exactly what most of you guys did for spring break. Probably what I did, right, was either you worked or you pursued leisurely activities. And I won't go into any more detail than that. All right, so let's go ahead and review. Today's lecture is going to be all about muscle tissue and muscle physiology. And I'll get to all this stuff here in just a little bit. This is going to be a fairly complex lecture. A lot of new terminology, a lot of new material is going to be thrown at you guys today. And we're going to be getting into the nitty-gritty molecular biology of muscle contraction as well. But let's start big picture, okay? So let's just do a quick review. What are the 3 general types of muscle? Well, we've got smooth muscle, we've got skeletal muscle, we've got cardiac muscle, right? You guys know that. You guys, some of you are good at identifying that on a slide, that histologically. So let's just review where the locations of these particular muscle tissues are in the body. So when it comes to smooth muscle, Okay, smooth muscle tends to be located within hollow organs, like your stomach, your bladder, your intestines, your lungs. You have smooth muscle in your lungs as well. Your blood vessels are hollow as well, right? They're hollow lumens filled with blood. Okay, so we see them in hollow organs, blood vessels, and we also see smooth muscle in the eye as well. And it is contraction and relaxation of smooth muscle in your eye that accounts for your pupil constricting and dilating. When your pupil constricts, for example, bands of smooth muscle contract, and that causes your pupils to get smaller, and then when they relax, they get larger. Does that kind of make sense? Well, when one muscle constricts and another relaxes, they get bigger and smaller. And we'll talk about how muscles tend to work in pairs or groups. Smooth muscle's a little different, but okay. The speed by which these work, smooth muscle tends to be a little slower, a little bit more leisurely in how it works. And when I talk about control, what I mean is, do you generally have conscious control over the muscle? Well, in the case of smooth muscle, we don't. We say that this is involuntary. Okay, this is smooth muscle contraction and relaxation largely tends to be out of our hands, out of our conscious control. Now, unconsciously, okay, involuntarily, there's a whole lot of stuff going on behind the scenes. We don't have to know that until we start studying neurology here in a few weeks. The cytological structure, so we look at the cytology of the cell structure. These tend to be small, okay? They tend to have a spindle-like appearance, like I drew here, and they only have one central nucleus. And when you get lots of these together, they look somewhat similar to stratified squamous epithelial tissue, right? They look very similar to that, but these are not flat like squamous epithelial tissue, rather these are kind of spindly shaped, okay? And then we move on to skeletal muscle, okay? Where is skeletal muscle located? Well, it's located generally, it's located on our bones. It's attached to our skeleton, our appendicular skeleton or axial skeleton. And muscles tend to be attached to bones via tendons, right? Tendons attach muscles to bones. And then, of course, ligaments attach bones to bones. Now, you do have some other ways that muscle can be attached, okay? You can have tendons, but you can also have something known as an aponeurosis. Okay? An aponeurosis is a flat tendon-like sheet, okay? So a tendon tends to be kind of a round, thick structure, right? And aponeurosis is kind of a flat sheet of connective tissue. And one of the primary areas that we would see in aponeurosis is actually in the abdomen. Certain groups of muscles in your abdominal wall come together, okay, are held together by connective tissue by an aponeurosis in your abdominal wall, and they're not necessarily attached to muscles via tendons. Okay, so this general rule of thumb tends to fall apart. Oh. How you guys doing?
Good.
You have a good spring break? Yeah? Yeah? All right. Nice and leisurely, relaxed? All right. Okay, that's great. You had a good spring break as well? Okay, okay. Well, it's over with now. Okay. Okay, good. All right. All right. So, skeletal muscle. Skeletal muscle, its speed tends to be rather quick, right? Tends to be rather quick acting. Okay. And in general, we tend to have voluntary control over skeletal muscle. That is, we have conscious control over it for the most part. Okay? When we look at skeletal muscle, it has a highly characteristic appearance to it, right? The cells themselves are long, they are straight, they are multinucleated, right? And they have the nuclei tend to be on the periphery of those muscle cells, or those muscle fibers is another name for a muscle cell, okay? And unlike the smooth muscle, skeletal muscle is striated, right? It has striations, okay? And then finally, cardiac muscle. And where is the only area in the body we find cardiac muscle?
The heart.
The heart, yeah. Okay, that's easy. And the speed of the heart can kind of vary. It can be very slow, it can be very fast. It has a lot of variability in it. And as you might guess, the control tends to be involuntary. Okay, so your heart contracting tends to be out of your conscious control. And heart cells, or myocytes, as we call them, okay, these have a characteristic appearance as well. They tend to be branched, right? They are also multinucleated. They do not have as many nuclei as your skeletal muscle cells have, okay? They tend to only have 1 or 2 nuclei per cell. They're branched. They have structures known as intercalated discs in between the individual muscle cells, the myocytes or the heart muscle cells, and those intercalated discs have a sodium channel in them that allows for the communication of sodium ions through that channel. And we'll talk about why that's important, or what's going on there. And like skeletal muscle, cardiac muscle is striated, has that robust striated pattern. Have I told you guys anything that you don't know at this point? Hopefully not, right? Hopefully this is just a review. So what I'm going to do now is I'm going to group 2 of these together because they share so many similarities. And which of these 3 types of muscle tissue tends to be more similar, if you had to guess? The skeletal and cardiac. Yeah, the skeletal and cardiac because they're multinucleated, and most importantly because they are striated. Okay, and remember, structure or form, okay, form informs function, right? Or function follows structure, structure follows form, however you want to put that. So clearly there's something curious about this presence of a striated pattern in these types of muscle. So what I'm going to do now is I'm going to move on from the review, and we're going to start talking about some new material. Some of this material we've actually talked about peripherally in lab already, at least some of the terminology. So this shouldn't be too unfamiliar with you guys. And if you've done your reading before coming to class, none of it should be unfamiliar, but I'm rather skeptical as to how much reading actually gets done outside of class. But maybe that skepticism is not warranted, but we'll see. All right, so first of all, in general, how do muscles work? Well, they contract, right? And when a muscle contracts, does it tend to get longer or shorter?
Shorter.
They tend to get shorter. Okay, that's the rule of thumb. They tend to get shorter when they contract. Okay, and there are some different types of muscle contraction that we want to talk about. You can have contraction without movement. Okay, what? Do you guys want an example of a contraction without movement? Okay, so let's say I put my hand against this wall and I push against it, right? Are muscles contracting? Am I contracting muscles? Are they contracting to push against the wall? Is chemical energy being converted into kinetic energy and is applying a force to this wall? Is that happening? Is the wall moving really?
No.
Not really, right? So there you go, right? So I have muscles contracting, but I don't actually have any notable movement going on. So when you have contraction without movement, we call that isometric contraction. So isometric contraction is a contraction where you don't really have any significant movement occurring. Okay? Now, isotonic contractions, on the other hand, are contractions that result in movement or motion. Okay? And there are 2 types or 2 flavors of isotonic. There are eccentric and concentric contractions. Okay? Eccentric is where you actually get contraction with lengthening, which is like what? What? That's not the general rule of thumb, is it? Okay, but it does happen, and we'll talk about that down here in a little bit. And then you get your classic isotonic contraction, and that is contraction with shortening of a muscle. Okay? So when a muscle lengthens, okay, it gets longer with the contraction, we call that eccentric, and when it gets shorter with the contraction, we call that concentric. And any movement in general associated with contraction is what we call isotonic. You guys cool with that? Does that kind of make sense? All right. So, how does this work? Okay. Well, first of all, what we need to understand is that muscles tend to work in pairs. Okay? They tend to work in pairs. Okay? And when we talk about a pair of muscles, okay, the first thing you need to ask yourself is, well, what kind of movement do I want to happen? Okay? And so what I'm going to do is I'm going to give you guys a really good example of a pair of muscles. You have your biceps brachii. Everyone knows where your biceps brachii is, right? It's right in here, right? It's on top of your humerus. Okay? Now, this tends to work with another muscle called your triceps brachii, which is back here. Okay? Now, when we assign terms to these muscles, we need to understand what's going on. Okay, so what I'm going to do is, I am going to look at the biceps and triceps in terms of, okay, in terms of flexion. So I'm going to flex my forearm, okay? And when I flex my forearm, okay, there is going to be a muscle that is the prime mover or the prime cause of that flexion, okay? And if you guys had to guess, which muscle would that be? That would be the biceps. Does that make sense? And so what we do is we say that the prime mover in whatever motion that we're looking at is the agonist. So when it comes to the flexion of my forearm, right? The flexion of the forearm, the prime mover, the agonist muscle for that would be the bicep. Does that make sense? Why would we call that the agonist, the prime mover? Okay, you guys cool with that? Now, you guys remember the difference between flexion and extension, right? Okay, flexion, okay, has to do with what? What are we doing when we flex?
Decreasing the angle.
Right, an angle is decreasing, right? So when I flex my forearm, right, the angle between here and here is about 180 right here, right? And then when I flex it, that angle decreases. That's about 90 there, right? Okay, so that's flexion. When I extend, the angle increases, right? So if I go from 90 to 180, that's extension, right? You guys remember that from lab. Flexion, extension, right? You guys make sense, right? Okay. All right, but we're talking about flexion, okay? So the agonist, okay, the muscle that causes the prime mover of that is the biceps brachii. Does that make sense why we call it the agonist for that kind of motion? You guys cool with that? Now, there are two other terms that can be a headache, can be an absolute headache. And these terms are known as insertion, okay, and origin, okay? And you're going to have to, in lab particularly, identify the origin and insertion of a muscle. So how do we keep this, what's going on here? How do we keep this straight? Well, just think of it this way, okay? Just think of it this way. The area where a muscle is fixed and not moving, okay? Where a muscle is fixed and it's not moving is what we call the origin, okay? And in general, the origin tends to be more proximal. It tends to be a proximal location, okay? And where the muscle does the moving is what we call the insertion. Okay? So when we look at the biceps, remember, the biceps is going to get smaller, right? It's going to contract. So when my bicep here gets smaller and contracts, okay, where do you suppose the origin is? Origin is up here, somewhere on the humerus, right? Does that make sense? It originates in the humerus? And the humerus really isn't moving, right? What's moving? The forearm is being moved, so where do you suppose the insertion is going to be? Yeah, it's going to be somewhere here in the forearm, right? So the origin, okay, the bicep is attached here. It's not moving here, right? When it gets shorter, and look, you can actually see my bicep shorten. You see that? It's not a very big bicep, but you can see it become shorter, right? Where is that movement occurring? That movement is distal to the point of insertion, right? And so clearly, or to origin, so clearly the insertion point is going to be down here somewhere in the forearm. Does that make sense? Right? So wherever the movement, okay, whatever's being acted upon and being moved, i.e. the forearm in this case, will be where the insertion is, and wherever it's not moving and it's attached and steady is what we call the origin. You guys cool with that? That's the difference between origin and insertion, okay? So the origin, okay, of the biceps brachii is going to be up here on the humerus, and the insertion is going to be down here in the forearm. You guys okay with that? Makes sense? Okay? So far so good? Okay, so when we talk about flexing the forearm, the agonist, the prime mover, is going to be the biceps brachii, okay? But remember, muscles tend to work in pairs, right? So the other major part of that equation is a muscle that wants to oppose that. And what muscle do you suppose is going to oppose flexing? The triceps, right? And if you think about it, if the triceps contracts, what happens to the forearm? It does what? It extends, right? An extension of the forearm is in diametric opposition to flexion, right? So the triceps and the biceps brachii are the pair, right? When we talk about flexion, the biceps would be the agonist, and the triceps would be the antagonist, right? They antagonize that motion. Does that make sense? Could we also say that when it comes to extending the forearm, the triceps would be the agonist and the bicep would be the antagonist? Does that make sense? So what is an agonist and what is an antagonist depends on the kind of motion that you're looking at. You guys okay with that? We cannot say that the bicep brachii is always going to be an agonist muscle because it may not. It depends on the motion that we're taking into consideration. You guys okay with that? Okay. Okay, so we talked about origin, we talked about insertion, we talked about agonist, antagonist, and then there are two other types of muscles that come into the equation sometimes. And these are what we call synergist muscles and fixator muscles. Okay? And a synergist muscle is a muscle that helps the agonist out. Okay? It helps the agonist out. Okay, it's not necessarily the prime mover, but it's still there and it helps out. Does that make sense? And the fixator muscles, these are muscles that tend to stabilize joints while the joints are moving. They're not necessarily involved in the motion itself. They're involved in keeping that joint stable. And where do you suppose, what areas of the body probably have lots of these stabilizing muscles, these stabilizer muscles? Shoulder, right? Because really the shoulder, where is the only place the shoulder is actually attached directly to the axial skeleton? The clavicle, right? It's the only place that it's attached. So you have to have a whole boatload of stabilizing muscle to keep that shoulder in place and stabilize it while the agonist and antagonist muscles are causing the motion that we want. Does that make sense? Okay. So you have those fixators. Synergist, a good example of that. Okay, so what is the primary muscle of breathing? The diaphragm, right? The diaphragm drops down, chest expands. But guess what? You also have little muscles in between your ribs called your intercostal muscles. You've got some muscles in your neck as well that help with expanding the rib cage out. And sometimes we call those accessory muscles to breathing, right? They're not the prime movers, they're not the diaphragm, but they help the diaphragm out nonetheless. So we would call those synergist muscles. Okay, you guys cool with that?
Is accessory muscle also a pretty common term?
Well, when it comes to breathing, yes. Okay, when it comes to breathing, absolutely. But synergist is the proper term for muscles that are helping the prime mover, helping the agonist out. You guys cool with that? So far, so good? All right, so now the next concept I want to talk about, again, we're big picture here, right? We're looking at the muscle big picture, okay? And just for reference, there are about 640 skeletal muscles, okay? And we won't even talk about smooth and that. But yeah, about 640 skeletal muscle, right? A lot of different muscle types. And each muscle is considered its own organ, right? And so when we talk about the musculoskeletal system, we're talking about many different organs, of course. Okay, so we're looking big picture right now, right? But what we'll do is we will reduce that. We'll reduce that down to the molecular level, more or less. Okay, now what I want to do is I want to talk about general types of muscle. You have two general types, and these are known as red and white fibers, or red and white muscle fibers. Okay, your red muscle fibers are called red because they tend to have a more reddish appearance to them. Okay, and what gives them that reddish appearance is a protein known as myoglobin. They have a large amount of myoglobin within them. They also have lots of mitochondria within them. Okay. And does anyone know what myoglobin does? What's the job of myoglobin? Does it sound somewhat similar to a very important protein that we've talked about before in this class? Kind of sounds like hemoglobin. Where's hemoglobin located? In your blood, right? In your red blood cells. What does hemoglobin do? Transports oxygen in your blood, so myoglobin just might. Yeah, so it's not so much a transporter in your muscles, but it is a container. Think of it as a storage area for oxygen. So when you are stressing that muscle out, okay, that muscle has oxygen stored within its myoglobin. So it allows that muscle to work longer. It has more endurance, okay? And we call these types of muscle fibers slow twitch, okay? They're not as fast moving, but they're in it for the long run. They're in it for the long haul, okay? So these are muscles that can work for a long period of time. Okay, for example, muscles involved in keeping me standing, right? I'm going to stand here for a couple of hours in front of you guys. Clearly, I need muscles that can continue to work for a long period of time. When I go out and I run, or when I ride my bike, okay, for hours on end, okay, clearly I need muscles that can make lots of energy, right? That can burn lots of glucose and convert a lot of fatty acids, okay, into ATP as well, okay? And so these are all slow twitch muscle fibers. These are the red fibers, these resist fatigue, and these are kind of your long haul muscles, or muscle fibers. Now your white fibers, okay, do not have as much hemoglobin, or myoglobin rather, so they fatigue much quicker, right? They fatigue faster. These tend to have a shorter duration, okay? And there are two subtypes of these type 2 or these what we call fast twitch muscle fibers. They're what we call intermediate twitch, and I think of those as kind of a combination. They're kind of in between full-on slow twitch and full-on short twitch. Okay, they're kind of in the middle. Okay, they can do some fatty acid stuff and they can do some anaerobic stuff, but they're not as robust as red fibers. And then your type 2B white fibers are your classic fast twitch. These are muscle fibers that are almost purely anaerobic. They don't have a whole lot of mitochondria, so most of their energy is anaerobically produced, which means they are going to fatigue very quickly, right? They're going to fatigue very quickly, and they're involved in fast, quick motions that you don't do over and over and over again. You guys okay with that? Okay, so these are your general types of muscle. Your red and white fibers. Your red are also known as your type 1. Your slow twitch. They resist fatigue. They've got a lot of myoglobin. They're in it for the long haul. Your white fibers tend to be shorter duration. They tend to have less myoglobin. They fast twitch. They fatigue quickly, and there are 2 subtypes. The 2A is more like the red fiber, is more on the red side, and the 2B is more on the white side. Okay. Yeah, so your 2A are what we call your intermediate twitch. Okay, so these are a little bit of red and a little bit of white. And then your 2B are more like your full-on white, your full-on fast twitch fibers. So far so good? Tracking like a pawn shop VCR. All right. Now, what I'm going to do is I'm now going to start talking about the anatomy and physiology of muscle, specifically skeletal muscle. We're going to start at skeletal muscle, and all of this is true for cardiac muscle as well. Okay, it's a very similar thing for cardiac muscle, and then at the end I will talk about smooth muscle because smooth muscle is very different and has some similar things going on, but they are a little different. Okay. So what I want you guys to do, and there are pictures in your book as well, okay? But what I want you to do is imagine that I've taken the bicep here, okay, the bicep brachii, and I chopped it in half. Okay, so here I have my bicep, I've chopped it in half. You can see the tendon here, right? The tendon that connects the bicep to either its insertion point or its origin. And I've cut through it. You guys cool with that? When we do that, we notice some anatomical features. First of all, we notice that the muscle is kind of like tubes surrounded by connective tissue, and then more tubes surrounded by connective tissue, and more tubes surrounded by connective tissue. Okay, so you kind of have a tube, connective tissue, tube, connective tissue, tube kind of construction to this. Does that make sense? And the tubes get smaller and smaller and smaller. Okay? So the first thing we notice is that the outermost part of the muscle is, like any organ, is going to be surrounded by a membrane, right? Every organ in your body is surrounded by a membrane. Muscle is no different, okay? And the outermost layer, okay, of connective tissue is what we call the epimysium, okay? The epimysium. The epimysium is the outermost layer of connective tissue. Okay, you cut through that. Okay, you cut through that, and what you see these bundles. Okay, so you see these large bundles, and then within them there are other little tubes. You see that there? Okay, these large bundles are what we call fascicles. These are fascicles. Okay. And the fascicles themselves are made up of another type of connective tissue known as perimysium. Okay? So epimysium is the outermost layer. Perimysium is what surrounds the fascicles. Okay? And then if I pull a fascicle out and I look at one of these fascicles, within the fascicles, okay, we're going to have yet another type of connective tissue called endomysium. And endomysium is the connective tissue that surrounds the actual muscle cells themselves. So, you have your muscle cell. Another name for a muscle cell is a muscle fiber, right? Remember talking about that in lab? Yet another name that may be used is myocyte. Myo means muscle, cyte means cell, right? Does that make sense? All right. So you could see either of these on a test: muscle cell, muscle fiber, or myocyte. They all mean the same thing. Okay? So you have your individual long muscle cells, and the muscle cells are surrounded by endomysium, and then the muscle cells are packaged into these larger units known as fascicles that are surrounded by perimysium, and then the fascicles themselves are surrounded by epimysium, and that makes up your entire muscle. Does that make sense? So you have several layers of connective tissue before you get to the actual muscle cells, the contractile cells themselves. You guys cool with that? And you need that connective tissue because you've got a lot of force being generated, right? And if you didn't have that connective tissue, those muscle cells might very well kind of explode and tear themselves apart, and it'd be kind of a bump deal. You guys cool with that? Make sense? So what I'm going to do now is I'm going to pull out, okay, so again, we started big picture and now we're moving down and we're kind of reducing things down. So I'm going to pull out an individual muscle fiber, and right here is an individual muscle fiber. And what I've done is I've peeled back, you can see the endomysium here. I've peeled back the endomysium and I have the actual muscle fiber itself. You guys cool with that? Okay, so there is some muscle-specific terminology when it comes to these muscle cells. Okay, we generally say that a cell has a plasma membrane or a cell membrane, right? That makes sense? But because muscle cells are so specialized, there's a special name for their membrane, and that is sarcolemma. Okay? Sarcolemma is a special term that means cell membrane. Okay? So sarcolemma just means the cell membrane of a muscle cell. You guys all right with that? Okay, sarcolemma equals cell membrane. Okay, it's just a cell membrane for muscle. Does that make sense? Okay. So you have this muscle cell membrane. Now within the muscle cell, so I cut the muscle cell open. Okay, within the muscle cell, I have these specialized structures known as myofibrils. Okay. Okay, myofibril is a specialized organelle within a muscle cell or myocyte. Okay? And they are suspended not in cytoplasm. Cytoplasm is just a general cell, but rather we call this sarcoplasm. Sarcoplasm is just muscle cell cytoplasm. Does that make sense? Again, because muscles are so specialized, we have assigned these special words. So sarcoplasm is just cytoplasm for a muscle cell. Okay? And within the sarcoplasm, you have these myofibrils. Okay? And I won't talk about that quite yet. Okay? But it's there. And you'll also notice something. You'll notice the nervous system itself. Okay. Specifically what are known as motor neurons. Okay, motor neurons, or these are sometimes known as efferent neurons. And an efferent neuron is a neuron that sends information away from the brain, right? If I want to move, if I want to pick up this bottle here, information needs to come from my brain into the muscles and make those muscles do what the brain wants, right? So that is descending or efferent or motor information, okay? So that information travels down these motor neurons And then these motor neurons are actually attached to the muscle cells. And they are attached at a special structure. And that special structure is known as, it has a couple of different names. Okay? It's either known as the neuromuscular junction, where the nerve and the muscle comes together, the neuromuscular junction, or another term is the motor end plate. Okay? It's the end of the motor neuron. Okay? So the neuromuscular junction or the motor end plate. Are you guys still following me okay? Mm-hmm. Okay, cool. So the information travels down the neuron, okay, and then it is transferred into the muscle cell or muscle fiber at the neuromuscular junction. Everybody's okay with that so far? Now what we're going to do is we're going to go deep. Okay? We're going to dig deep. What I'm going to do is I'm now going to enlarge this neuromuscular junction here, and that's what I've drawn right here. I've enlarged this. So what we have is we have the axon, right? Axons go away from neurons, right? I have the distal axon, okay? So this is the very end of the axon here. And in the distal part of the axon, I have little vesicles. You remember what vesicles are from talking about cytology, right? Vesicles are little membrane-bound spheres that contain chemicals within them. Sometimes they're enzymes, okay? Other times, as is the case with neurons, they contain a communicating chemical known as a neurotransmitter. Okay? So far, so good? And what happens is, okay, the neuron, okay, to get that signal to the muscle, the neuron has to do something very interesting. And remember, toward the beginning of class, now we're combining multiple levels of knowledge here. We said that there is a special protein called the sodium-potassium pump, right? Mm-hmm. And what does the sodium-potassium pump do?
It pumps the sodium out and brings the
potassium in, okay? So the sodium-potassium pump, okay, is active transport. It pumps sodium out of the cell and pumps potassium in. So where is most of the sodium located in your body? In or out of the cell? Out of the cell, right? Where do you suppose that sodium really wants to go? Into the cell, right? But it's pumped out. Now to get that, transmit that information, what happens is the neuron undergoes a process known as depolarization. Depolarization. Okay? And what happens is sodium will rush into the cell, right? Because normally the sodium's outside, right? So normally it's positively charged outside compared to inside the cell. Does that make sense? Okay, so normally the cell is more negatively charged inside than out. So depolarization means that the cell becomes less negatively charged because all that sodium rushes in, right? So sodium rushes in to this neuron, and that depolarization travels down the neuron in a wave. So as it travels down the axon, the sodium's rushing in, and it depolarizes, okay? And that wave of depolarization goes all the way down the axon, okay? The sodium is rushing in until it gets to the end of the axon. And then there are some things that happen. We won't talk about them today because we'll talk about them in a lot more detail in neurology. But ultimately, when that wave of depolarization gets to the end of the axon, it causes these vesicles that contain neurotransmitters, it causes these vesicles to leave the neuron. It causes them to undergo exocytosis. And they leave and they fuse with the distal end, and then they open up, right? Once they fuse, they open up and they dump. They dump their neurotransmitters out into this space. That neurotransmitter gets dumped out into this space between the neuron and the muscle. You guys okay with that? And that space that separates the neuron and the muscle is known as the synapse. Okay? You guys okay with that? Okay. Now, you will need to know the name of the neurotransmitter that is released from the distal end of a motor neuron. And it is one of the most important neurotransmitters in physiology. And that neurotransmitter is known as acetylcholine. Acetylcholine. It's basically acetic acid, vinegar, attached to a choline molecule. So it's a very simple molecule. Okay? Acetylcholine. Acetylcholine is released from this neuron and it diffuses into the synapse. Okay? And oftentimes acetylcholine is abbreviated by an ACH. So when you see ACH, that means acetylcholine. You guys okay with that? That acetylcholine gets released into the synapse, and then there are little receptors. There are receptors, okay, here on the sarcolemma of the muscle cell. Remember, the sarcolemma is the cell membrane of the muscle cell. There are receptors. There are receptors that this acetylcholine attaches to. And these are known as nicotinic acetylcholine receptors. Nicotinic. Kind of like nicotine, right? In a cigarette. Okay? These are nicotinic acetylcholine receptors. And what happens is, okay, the acetylcholine attaches to those receptors, and those receptors are actually what we call ion channels. They're little gates that are closed. And when the acetylcholine attaches to those gates, it causes them to open up. You guys okay with that? Right? They're a receptor, there's a conformational change, they open up. And what they do is when they open up, they allow sodium to rush into the muscle cell. So that does what to the muscle cell? It doesn't contract. Not yet, at least. Well, when sodium's rushing into the neuron, we say that it is depolarizing, right? So when the neuron dumps a bunch of acetylcholine onto the muscle cell, and those receptors open up, those nicotinic receptors open up, and sodium rushes in, the muscle cell undergoes depolarization, right? The muscle cell depolarizes. Those little channels open up, and all that sodium starts rushing into the muscle cell and depolarizes. So basically you have depolarization, boom, neurotransmitter tells the muscle to depolarize. Make sense? You guys okay with that? So at this point, the muscle cell depolarizes.
So the ACH opens the gates, but the sodium goes in.
Yes.
Okay.
Yeah, so remember, you have sodium potassium pumps, all the sodium's pushed out of the cell, and then, but once that acetylcholine starts attaching to these nicotinic receptors, they start popping open, and then sodium starts rushing into that muscle cell. And so the muscle cell begins to depolarize. You guys cool with that? So far, so good? We haven't even got to contraction yet.
The ACH will not open until they're attached to the nicotinic.
Well, ACH doesn't. ACH is a molecule that causes the receptor to open. Right? ACH is the molecule, is the neurotransmitter released by the neuron, right? It tells those channels to open. causing the sodium to rush in. Right? Just think of the channel as a door. I have this door here. Right? I have this door here. Okay? And this acetylcholine opens the door. Right? And then as long as there's enough sodium, the sodium will be able to diffuse through the door. Does that kind of make sense there? How that kind of works there? If you can kind of think of it like that, and that's a really good model for what's going on. Okay, so far so good? All right, we haven't even got to contraction yet, have we? This stuff is a little tough. Okay, so what I'm going to do before we get to contractions, we've got to talk about some more terminology, all right? And then I'll put you guys on a break because it's almost been an hour already. Can you believe that? Okay, so what I'm going to do now is, you see this myofibril here, right? This myofibril, okay? I'm going to pull one of these out and I'm going to enlarge. I'm going to look at it, okay? And we notice that we have little striations on the myofibrils here. And when I enlarge this, okay, I see a structure that looks like this, okay? I see a structure that looks like this. I see these dark lines, okay, and these dark vertical lines here, are what cause those striations there, okay? And because they kind of have like a little Z-like appearance to them, we call these the Z lines. You guys okay with that? Z lines are vertical, okay? They are what cause that striated pattern. So far, so good? Okay. And the functional or the contractile unit of a muscle cell, okay, an individual contractile unit, and you have thousands and thousands of these within an individual muscle cell, but the individual contractile unit is something known as a sarcomere. Okay, a sarcomere. And a sarcomere or a sacromere, however you want to announce that, a sacromere is the unit between one Z line and another Z line. So you see that in between two Z lines? So here's a Z line, here's a Z line, here's a Z line. So Z line to Z line is a sarcomere. That's not a cell. It's not a cell. Okay, that is a small unit within an organelle of a cell, within the myofibril of the cell itself. Okay, so a sarcomere is a subcellular structure. You guys okay with that? It's the fundamental unit, contractile unit. All right. So Z line to Z line is what we call a sarcomere. Now, these sarcomeres have two major structures within them. And these are little line or filament-like structures. Okay? They have these thin filament structures here known as thin filaments. All right? So far, so good. All right? And these thin filaments are made up of a protein, or primarily made up of a protein known as actin, or it's actually several different molecules. And we'll look at it in a little more detail here in a bit. You guys good with that? So your Z lines and your thin filaments are made up of actin, and then in between the thin filaments, you have these thicker structures composed of another protein known as myosin. And as you might guess, the myosin, because it's thicker looking, is what we call the thick filament. So are you guys okay with thin filaments and thick filaments and Z lines? Okay? Now, to make it even more complicated, what you have is you have different zones and bands within the sarcomeres. Okay? And so let's start at the H zone first. Okay? The H zone, see this little opening here in the sarcomere? See that opening there? You guys see that okay? The only thing you have in that opening is what? Just myosin, just a thick filament, right? That's the only structure you have in that opening. So the H zone is myosin only, okay? Does that make sense? Okay. Now the I band, on the other hand, is only what? Thin filaments, right? So the I band is only thin filaments, okay? So you've got your H zone, thick, I band, only thin, and then you have your, what we call your A band, and your A band is the entire thick filament, right? So from one end of the thick filament to the other end. But does the A band also include some thin filament? Yes. Okay. So the H zone is all thick filament, your I band is all thin filament, and your A band is a combination because it is the entire length of the thick filament. You guys okay with that? With those structures there? Okay. So at this point, we now have the fundamental foundation, the foundational material that we need to understand what's going on with contraction, but that is something that we'll pick up in a few minutes. How about you guys take a quick 5-minute break, and then we'll come back and we'll hurt our brains some more.